GPCRs are receptors in our body that detect signals like taste, smell, and vision. They respond to different molecules like neurotransmitters or flavors. When the right molecule binds to the receptor, it triggers a response that helps us perceive things like taste or smell. Essentially, these receptors help us experience the world around us. All G-protein coupled receptors, or GPCRs, share a conserved and elegant architecture. They thread through the plasma membrane exactly 7 times, forming 7 transmembrane alpha helices, known as H1 through H7. Each GPCR includes 4 extracellular loops and 4 cytosolic loops, with the N-terminus facing the exoplasmic side and the C-terminus facing the cytosol. This orientation is not random, it's evolutionarily conserved. What anchors these receptors so firmly into the membrane? The answer lies in hydrophobic amino acids coating the outer surfaces of those helices. These residues interact stably with the fatty acid tails of the lipid bilayer, locking the receptor into place. In the resting state, the G-protein coupled receptor, or GPCR, is inactive, and nearby a heterotrimeric G protein composed of alpha, beta, and gamma subunits remains tethered to the plasma membrane. The G-alpha subunit binds GDP and is associated with the G-beta gamma dimer. Both G-alpha and G-gamma are anchored to the membrane via covalently attached lipids. When a hormone binds to the extracellular domain of the receptor, it causes a conformational change that extends to the cytoplasmic side. This activated receptor then binds to the G-alpha subunit, triggering the release of GDP. Acting as a guanine nucleotide exchange factor, or GEF, the receptor facilitates the rapid binding of GTP to G-alpha, which causes a change in its conformation. As a result, G-alpha GTP dissociates from both the receptor and the G-beta gamma dimer. Now, both G-alpha, GTP, and G-beta gamma can independently interact with effector proteins. In this example, G-alpha GTP activates the effector, initiating a signaling cascade. This active state is short-lived. The intrinsic GTP-ACE activity of G-alpha hydrolyzes GTP to GDP, inactivating the subunit. Finally, G-alpha GDP reassociates with G-beta gamma, restoring the inactive trimeric G-protein complex and resetting the system for another cycle of activation. In GPCR signaling, all effector proteins are either membrane-bound ion channels or enzymes that produce second messengers. Second messengers are small molecules like CAMP, IP3, DAG, and calcium ions. They're produced inside the cell when GPCR signaling is activated, and they play a key role in amplifying the signal and triggering downstream signaling pathways that shape the cell's response. The diversity in signaling comes from different G proteins, each defined by its G-alpha subunit. Humans have 21 types of G-alpha subunits, which determine the specific cellular response. For example, epinephrine binds to different adrenergic receptors. Beta-1 and beta-2 receptors activate GS, stimulating adenylyl cyclase and increasing CAMP. Alpha-1 receptors couple with GQ, activating phospholipase C to produce DAG and IP3. Meanwhile, A2 receptors use GI to inhibit adenylyl cyclase, reducing CAMP levels. GPCRs are involved in everything from how we see and smell to how our cells respond to signals, and this is just the beginning. I'm creating a playlist that walks through different parts of GPCR signaling, so if you're into this kind of cell talk, stick around. There's more on the way.